Hi, my name is Reich, and in this video I will be presenting on a figure from a paper entitled The Role of Tau in the Pathological Process and Clinical Expression of Huntington's Disease. Before I dive into my figure, I want to start with some background information on the tau protein. Tau is a protein encoded by the MAPT gene that's involved in many important cellular functions. These include microtubule assembly, stabilization, neuronal morphogenesis, and axonal transport, all which are modulated by site-specific phosphorylation. Alternative splicing of MAPT exon 10 results in isoforms with either 3 or 4 carboxy-terminal microtubule binding repeat domains. I included this image to make it easier to visualize. You can see the microtubular binding domains, on which there are 4 repeat domains in this image. These 3R and 4R domains are expressed in approximately equal amounts in the central nervous system of healthy adults. It's been previously found that mutations in common genetic variants of the MAPT gene have been associated with the development of tauopathies, as well as imbalances in the 4R to 3R tau ratio isoform, which may contribute to tau hypophosphorylation and aggregation. So how does tau relate to Huntington's disease? Recently, it has been shown that there is an increase in the 4R to 3R tau isoform ratio in Huntington's disease post-mortem brain samples. Subsequently, it has also been shown in transgenic Huntington's disease mouse models that mutant HTT alters tau phosphorylation, resulting in insoluble neurofibrillary tangles. Based on this background, the researchers set out to answer the following question: do tau aggregates and their level of phosphorylation affect the expression and/or progression of Huntington's disease? In order to answer this overarching question, the authors focused their research around 3 sub-questions. How does tau aggregation in Huntington's disease compare to tau aggregation in other neurodegenerative diseases and healthy controls? Is tau aggregation truly linked to Huntington's disease and not just old age? And what is the relationship between MAPT haplotypes and the progression of Huntington's disease? My figure explored parts of these first and second sub-questions by studying the tau pathology of Huntington's disease. Figure 1 shows various types of tau pathologies observed in Huntington's disease brains. This figure was generated through multiple experiments using immunochemistry on paraffin-embedded sections of Huntington's disease brain samples. Immunochemistry is the use of antibodies and color change to identify protein concentration in tissues. In this case, antibodies were used to identify tau deposits. In the next 3 slides, I'm going to focus more narrowly on what is observed in panels A, C, and D. These 2 images show tau aggregation in both cortical and striatal brain tissue samples with Huntington's disease. The arrows and arrowheads point to different morphologies of the tau deposits, such as flame shape, the red arrow, and globular, the red arrowhead, to highlight a few. For reference, the scale bar in the larger images represents 50 micrometers, and the scale bar on the smaller insets reflects 20 micrometers. These images show phosphorylated tau aggregates from patients with young-onset Huntington's disease. The image on the left is from a patient who died at 26 and the right at 40, both from Huntington's disease. This image shows oligomeric tau inclusions in striatal Huntington's disease tissue, which are the numerous orange shapes you see in these images. So this experiment was able to confirm and extend recent findings by reporting pathological inclusions containing abnormally phosphorylated tau protein in the cortex and striatal tissue of human Huntington's disease brains. They were also able to confirm that tau pathology in Huntington's disease occurred independently of the patient's age. Furthermore, tau oligomers were observed, which are considered to be the most likely neurotoxic tau deposit. Most importantly, these results further reinforce the notion that the presence of tau in the brains of patients with Huntington's disease is of true pathological importance. The overall conclusion of this paper is that these experiments show the novel role of tau in both the pathological process and clinical expression of Huntington's disease. The results shown in Figure 1 focus on the pathological aspect of the conclusion by highlighting the implications of tau in the development of Huntington's disease. This paper contributes to the field by emphasizing the role of tau in the pathogenic process and clinical expression of Huntington's disease, which in turn opens up new therapeutic possibilities for this incurable condition. Thank you for watching, and I hope you learned something new.